Hello, this is Dr. James Camp from Lee College in Baytown, Texas. And if you're taking one of my anatomy and physiology classes, or if you're taking anatomy and physiology anywhere, you're probably going to have to make sketches of microscope slides of cells or tissues. Now, a lot of people get the wrong ideas about how to approach one of these sketches. They either just want something that vaguely looks like the tissue they're looking at, or they want, or they get so focused on individual details that it takes them an hour to make every sketch. So I want to give a little bit of general advice here on how to approach a microscope sketch to get more credit, more points for your sketch, and at the same time not waste huge amounts of time on it. The first problem a lot of people have is sketching a single cell. You're rarely going to see a single cell in a microscope slide. This is a picture here of a human sperm smear. Okay, so it's a collection of cells. You're not going to be able to get one individual sperm cell on the slide and focus close up enough to get just that one cell. So how do you approach this? One typical student approach would be to say, okay, I see a little blue thing there, and a little blue thing there, and a little blue thing there, and a little blue thing there, 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 and I see a purple tail, purple tail, purple tail, purple tail, purple tail, squiggly purple tail, purple tail. I'm done. Oh, well, there are a lot of purple dots, so I'll go through and I'll do that too, and that'll just make my drawing look even more confusing, so that when my professor is looking at this to try to assign it a grade, they're going to say, what the heck does this person think they're seeing? The problem with this sketch, there are two problems. One is that if I were to just pick this up off the floor, I would not have a clue that what you just drew here was a collection of sperm cells. Okay, that could be a lot of things. Okay. But the second is that you've put the details in the wrong places. All these little dots that this person drew are completely distracting from the purpose of the slide, which is the sperm cells themselves. So let's go back and try again. I've seen that same bad approach, by the way, not just with the sperm cell slide, but with blood cell slides. I've seen a lot of people who their approach to drawing a blood cell slide is to put little red dots everywhere that they see a red blood cell, and then, okay, here's a white blood cell. Well, it's squiggly, and it's got a nucleus, so there we go. Again, not actually enough detail for me to know that you really recognized what you were looking at, and a lot of extraneous details, in putting in all of these dots that distract from the image. So let's say we want to focus on one little sperm cell. Maybe we want to focus on this one right here because it's got a lot of detail to it. Okay, so I'm going to come here and I'm going to say, okay, I can see that the nucleus of the sperm cell is pretty much a solid blue dot there. Okay, and then I'm going to take my labeling pencil and I'm going to say nucleus. I'm going to learn to spell too. Okay. The next thing I notice is that right here there's a bulge just behind the head of the sperm cell. So I can capture that pretty well with a couple of rounded lines and fill in with some little bit of detail there. I see some dots within this. Okay, and we know from electron microscopy that what all those little dots you're seeing in there are actual mitochondria packed into this mid piece, we call it, the power plant of the sperm cell, but we don't know that from this image, so I'm not going to try to draw it. Then I see that the tail is a fairly long, thin line. So I'm going to draw that like that. Okay. That's all the detail you really need for capturing a sperm cell, but it's much more obvious now. And I go back with my labeling pencil, usually in a different color, and I say that this is the mid piece, and this is the tail. Okay. And it's usually, at this point, if I picked that slide up off the floor, that sketch, it would be very obvious to me that what you had just drawn is a sperm cell. Okay. I mentioned blood cells as well. How would I approach that? Well, I would draw a couple of red blood cells for size and scale, and to show that they are present on the same slide as my white blood cell. And then I would take my white blood cell and I'd draw it at about the size that it showed up compared to the red blood cells. And I would draw its nucleus, which in this one that I'm imagining has 4 lobes. Okay. And then I would notice that this one has orange dots, or granules we call them, in its cytoplasm, so I would draw those in. I wouldn't feel like I have to draw every granule that I see on the slide. I'd just draw enough of them for it to be obvious that that's going on. Okay, and then with my different colored labeling pencil, I would note that that's an erythrocyte and that this is a neutrophil. Okay. Notice that I'm not restricting myself to drawing these at exactly the same size scale that they showed up on the original microscope image. The sperm over here I enlarged somewhat. The red blood cell and white blood cells here I'm enlarging somewhat. That's typical practice when you're making sketches of a single cell in a microscope slide, is that it's okay to enlarge them a little bit so that your hand can capture the details that your eye is capable of seeing. Okay. How about sketching a tissue? Now this is a fairly difficult tissue to sketch here that I picked, and I did that on purpose. This is elastic cartilage, and you can see there are a lot of things going on here. There are individual cartilage cells, chondrocytes, okay? They are surrounded by these walled spaces called lacunae, and in between them we have purple elastic fibers running through the cartilage. Okay. So what do we do with this? And the cells get flatter as they get towards the edges of the tissue, and you even change from a dark blue to a sort of a pinkish color of the extracellular matrix around the cells. So what I'm going to do is I'm going to, again, kind of enlarge a little bit. I'm going to pick this area to zoom in on, and I'm going to say, okay, let me pick on just this box to start with. I see a cell. It's got a weird little white area in it. Maybe that's its nucleus. It's got dark blue around that, and then everything else is kind of uniformly purple. Then there's kind of a halo around that cell, and then there's a purple wall around that, and there's a blue wall around that, and then immediately below that there's another blue wall with another purple wall inside it, and another one of these kind of pear-shaped purple cells with a white circle in it surrounded by a blue circle, and then everything else is pretty much purple. Okay, so now I've captured those two squares in some detail, and now I can show that there is another cell like this up here and another cell like this right here. Okay, and they both got that purple surrounded by blue going on. And then I can draw that there are elastic fibers running through this area. Okay, if I have time, I can go back in and fill in the actual cells that were in these spaces. And I can see that down here these fibers fray a little bit, and there are places where they kind of crisscross a bit, and then there's another cell in this area, another lacuna, and another one kind of over here, and then there's an area of kind of crisscrossing fibers here. Okay, up here I start to get flatter cells. Okay, and the fibers kind of wear out there, and then I have these pinkish purple fibers running along the baseline here. And running along there. And that's a mess, right? Okay. But it captures a lot of what's going on without having to get into serious levels of detail. I could draw another cell here if I want to. Okay, and show that more of these purple fibers go around it, and then there's another cell space there, and another cell space here, and these purple fibers go around those two. Okay. Now the key is that I'm then going to go back in with my labeling pencil and say, okay, here is a lacuna, okay, singular of lacunae. Here is a chondrocyte. Here are elastic fibers, okay, and here I've got the perichondrium. Okay, now that I've labeled those things, my mess of a drawing is a little bit more understandable. And maybe you're a neater artist than I am and you can do this better than I just did. But there are three major rules that I followed in making this drawing here. Okay, one is draw individual cells, making sure to keep their shapes and their nucleus and their shapes, their nuclei, all of those things in proper proportion to each other. You can enlarge them a little bit, you can simplify the drawing a little bit, but be sure you are drawing individual cells. One of the common mistakes I see with this drawing is people say, okay, well, there's something that looks kind of like this, and then there are these fibers here. Okay, I'm done. Well, what is any of that? Okay, or with epithelial tissues, I often see, okay, well, there's a circle here, and there's a circle here, and there's a circle here. Well, each of those circles is made up of cells. And if you haven't drawn the individual cells that make up this circle, then I don't know, as your professor who's grading this, that you recognize that there were cells making up this structure. Okay? And you, when you look back at this as for studying purposes, won't be able to tell that there were cells making up that structure. Okay, so now that's obviously different from here, where the circles are lacunae around an individual cell. Okay, so you don't want to draw this made up of lots of cells. You want to draw that exactly as we drew it, as a solid line with a cell inside it. Okay. Second thing I did was preserve shapes and proportions. Okay, I can zoom in on things a little bit. I can magnify this. I can have it so that it's only a couple of cell layers high instead of 6 or 7 cell layers high, as long as I'm preserving the shapes of things and the relative proportions. Okay, that they get smaller as they get toward the edges in this case. Another thing I'm doing is I'm noting what we might call textures and colors. Okay, so the colors in this case mean something. If it's a dark blue, it's an elastic fiber. If it's more of a purplish color, it's a cell. If it's that pinkish color at the edge, which I didn't have a pink to do, so I made it purple, but that's the collagen fibers of the perichondrium. Okay. So, maybe I should have made this red to show that it's collagen fibers. And I noticed that the texture of these elastic fibers is that they are stringy and squiggly. They don't go straight. They crisscross, they squiggle. Okay, so that's an important thing to note about about elastic fibers. Okay. Finally, what if you're sketching a multi-layer tissue? In this case, you've got too much detail to afford to draw every single cell exactly the way it is, right? Well, maybe, maybe not. Again, a very common mistake I see with this is somebody draws, okay, well, there's a hole there, and there's a pink layer around it, so I'm just going to fill that all in with pink, and then there's a dark red layer around that. Pick the same red there. Let me pick a different red for my pink. Okay. So I'm going to fill that in with red, and I'm going to just fill that in with red, and see, it's a 3-layer tissue, and then there's a speckly area in the middle here. Okay, I've captured all the layers, right? Well, yes and no. You haven't told me anything about the layers by the way you drew them. Okay, so what I want to know, again, think about the perspective of your science teacher here. Your science teacher wants to know that you understood what you were looking at, not just that you can draw circles. So here, if I look closely, this inner circle is actually made of lots of little flat cells. So my first rule from the previous page is draw individual cells and preserve, cell shapes. So there we go. We've got individual cells there, and my labeling pencil, I can go over here and say endothelial cells. Okay, then if I look, there's a bright layer. I don't have the right color for it, but there's a bright layer that squiggles around this before we hit the pink. And with my labeling pencil, I'm going to call that the elastic lamina, because I happen to know that that's the name of that thing. Okay, then I'm going to take whatever color I've got for pink, and I'm going to draw these spindly shaped cells all the way around. Okay. And I may need to go in, with my microscope image, zoom in further to see what this is all made of so that I can draw these right. Okay. And with my labeling pencil, I'm going to indicate that these are smooth muscle cells. Okay. And then I don't have to keep, and I can give some of them a little nucleus to show that I know they've got a nucleus in there. Okay. Once again, I can simplify a little bit as long as I maintain the shape and the pattern. Okay. And then go back and give some of those nuclei. Okay. And then if I'm tired of drawing but I want to indicate that that goes on for a little longer, I can indicate with dots that I know that this, with dashes, that I know that this area is about that big. Then I get my brick red, and I know that this is made of collagen fibers. Okay, so I'm going to draw bigger, longer squiggles here. And with my labeling pencil, I'm going to indicate collagen fibers. Okay, and then I can go back with my labeling pencil and I can say that this is the tunica interna, this is the tunica media, and this is the tunica externa. Okay, and now I know exactly what I'm looking at when I look at that picture. I have everything labeled. I have everything drawn in a kind of exaggerated way so that I can see what everything is made of. And I have everything labeled so that when I go back to look at it later, I know what I was drawing. Okay. So that wraps up some of my advice on sketching. I hope that was helpful to you, and good luck with your own sketches.